Our first speaker today is Mukesh Verma, and he's in the DCCPS. He's chief of the Methods and Technologies branch. He got his Ph.D. in India, and then he was at George Washington University, and then he was in the Division of Cancer Prevention, where he led the Small Business Innovation Research Program. And then he joined the EGRP as a program director in 2004. And he's currently the program director, Epidemiology and Genomics Research Branch. His title, Epigenetics and Cancer Control and Prevention. Are we ready for the prime time? Mukesh.
Thanks a lot for giving me this opportunity to talk about epigenetics. As the title indicates, I will go and explain about where we stand in terms of using epigenetic approaches in cancer control and prevention. So just to introduce this topic, epigenetics, whenever gene expression changes, but there is no change in gene structure, like in genetics, you know, mutation, deletion, addition, or SNPs will be there. So their sequence or DNA changes. But if it is not changed, still gene expression changes. It's called epigenetics. So genetic code in genetics, you know, but epigenetics has two major codes. One is called methylation code. Another is called histone code. Methylation happens mostly in DNA promoter region, where CG sequences, when they come, they are called CpG-RLAN, and that C cytidine get methylated at 5' end, and it's called hypermethylation, and which changes gene expression. In histone, several modifications are there, which I am going to discuss later on. They also affect gene expression. And altogether, there is a balance of methylation and histone modifications. So in general, if you want to know, genetics tells us what we are capable of, what we can do. But epigenetics will tell when we should do something and when we should stop. So as I mentioned, there is a balance of methylation code and histone code. Time to time, you will see interesting or those articles which distinguish genetics with epigenetics, or they will tell like why your DNA isn't your destiny. So studies were conducted in monozygotic twins where genome is the same, but still discordance was found in twins when they were developed. And it was found that due to promoter methylation and histone modification, one of them developed cardiovascular disease, another did not develop. And that's how they showed that genetics and epigenetics are different and they are distinguished. And based on that, many things came later on, like pharmacoepigenomics and pharmacogenomics, or genome-wide association studies and epigenome-wide association studies. So in cancer, as you know, that cancer affects almost all organ sites, and prostate cancer in men and breast cancer in women, that is the most highest fatal one, where incidence and mortality is high. In other cancers also it happens, and it is estimated that by 2022, about 18 million people will be affected by cancer. In our division, which is called Division of Cancer Control and Population Sciences, we cover all cancer continuum, starting from prevention, how we can prevent that, or early detection, or diagnosis, and after treatment also, what is the quality of life, how we can improve that, and end of life, after mortality also we keep data. That CR data, which is called surveillance epidemiological data, that comes from our division. So cancer prevention can be at two places, like cancer recurrence to stop that, or stopping secondary cancer. And prevention we defined either restoring transcription or halting progression and stopping metastasis. What we emphasize in cancer, in our program especially, These cancers are the major cancers, but they take long time to develop from the initiation up to fully developed. So we want to emphasize on those stages which are at early stages of cancer development. And idea is that if we identify some biomarkers, then we have some option for treatment or controlling that cancer. In genetics, it has been shown, like in APC gene mutation or RAS gene or PI3K, they were in those colorectal cancer, they were identified. Similarly, now in epigenetics also, we know few markers which can be used for early detection. Here it is again shown that how many years and after what age these start coming or showing some symptoms, and based on that, subtyping or name of the cancer is given. SIN1, SIN2, SIN3, all those things are there for major cancers. Regarding epigenetics, one major thing you have to understand that for understanding genetics, you don't have to know epigenetics. But if you want to know about epigenetics, the genetic background comes first. So that is the main thing. And if you see how historically it was developed, so in 1850 to 1900, Mendelian inheritance, that you heard about that. Then 1900 to 1950, that time gene concept or mutation or genotype, phenotype concept came. And then from 50 to 2000 was structure of DNA, sequence of DNA, and genetic code that came. And after 2000, the epigenetics idea has come. And this genetics and epigenetics, they go side by side in gene regulation or cancer development. So in genome, we know that in different organ sites, mutations have been identified. And in fact, major pathways are here, which Vogelstein identified, I think 13 pathways, which regulate cell survival, cell fate, or genomic maintenance. And when genome-wide association studies were done, then for different cancers, at different loci, those mutations or SNPs were identified. So 2005 or 6 or 7, and these are different chromosomes in which different mutations were identified, and some were disease associated, some were not disease associated. But that way, this progress was done in genetics. And what we want to do, based on that background, we want to see how much epigenetic changes are there, because still some gaps were there. And when genome-wide association studies were done, where even single nucleotide polymorphism was identified, then many times those genetic changes were not in the gene region which codes for gene. And many times they were outside the gene. So then the idea came, for which we have one major program also. It's called Common Fund Program, and it's called 4D nucleome. So along with methylation changes and histone changes, nucleome structure also changes. Either it becomes compact or relaxed. So now what we are doing, that taking different images, the structure of chromatin is seen, and see which one is in heterochromatic form or which is in homochromatic form. One is active chromatin or repressive chromatin, which represent the active transcription. So unit of chromatin is nucleosome. It's about 146 nucleotide base pair long. And around DNA, these histone and non-histone proteins, they surround that to give it stability. And histones are of 4 kinds: H2A, H2B, H3, and H4. H1 is a linker. In 74, Roger Kornberg was awarded by Nobel Prize for nucleosome structure. And initially, I indicated that in promoter region, certain CG sequences are there, and when C is methylated, it is called hypermethylation. And whenever, this is the promoter region of a gene, whenever hypermethylation is there, then gene gets inactivated. Example is tumor suppressor genes. Tumor suppressor gene, in general, they function to suppress tumor, but if promoter is hypermethylated, then gene get inactivated. Similarly, sometime in the promoter region already CpG island or they are hypermethylated, then this methylation group is detached and hypomethylation happens, which is reported in oncogenes. So both ways, depending on what situation is there, they contribute to cancer development. Along with methylation and histone, long non-coding RNA are also part of epigenetic mechanism. which are like microRNA or long non-coding RNA. Methylation occurs by DNA methylation transferase. They are also of different kinds. Some are for initiation of methylation, some are for methylation. Similarly, histone is modified by ubiquitination, phosphorylation, methylation, acetylation. So different modifications are there. Few more terms are used in epigenetics. Like CG sequence, I told if more than 40% in the promoter region are there, these are called CPG Ireland. CG sequence will be there. Another term came for shorts if CPG Irelands are less than 2 kb from CPG Ireland, and then shells, they are located between 2 to 4 kb, and other region is called open C. So these are the terms. Here what I have shown that what are the factors which contribute to epigenetic regulation, and different cancer development stages, they are affected by epigenetics. About DNA methyltransferase, I told you. Histone modifications are by histone acetyltransferase or deacetylation. So in histone also, either acetyl group will attach or detach, and that also regulate gene transcription. Some proteins are histone proteins, some are non-histone proteins. One of the example is polychrome group of proteins. So they also affect chromatin compactation or relaxation. And similarly, another protein was also identified, EZH2. When they are in combination, then targets can be made for these proteins. And I will show you later on its clinical implication and how much it has been used. MicroRNAs are also part of this regulation. And you know, like in genome, we have ALU sequences or repeat sequences, like sign and line sequences, long interspersed sequences. So they are already methylated, and with age or with disease development, they get hypomethylated. So Peter Jones and Andrew Feinberg, they did initial work, and they showed in transcription, first this starts with acetylation, and in histone I mentioned S2A, S2B, S3, and H4. So S3K9 it starts or active chromatin, this histone is activated, then deacetylation of S3K9 is deactivation, and then completely silence when it is methylated. So if you see this part, then nucleosome, which are in chromatin, they are quite apart. Yellow color shows acetylation. So acetylation, it means gene is active because in between chromatin you have, in between nucleosome you have space. But if you come to this stage, gradually this red color shows hypermethylation. So deacetylation by removing acetylation and addition of methylation and compactation of chromatin, that causes the gene transcription inhibition. So that's how stepwise it happens. And nowadays, many more other proteins also have been identified. And also the sequence when some protein binds and after how much time it will bind, those have been identified. Whenever methylation experiments are done, then generally sequencing is done to determine methylation on both strands, because that is not all of none. One time it starts in one strand, then it starts in another strand. The way Vogelstein in genomic regulation, they propose like gatekeeper gene or housekeeping gene. Similarly, for epigenetic regulation, some modifications are called writers or readers or erasers. Writers are those which will put mark on DNA, epigenetic mark. Readers which will read those proteins which can recognize that. Eraser like deacetylase which will remove that. So this kind of terminology also you will see. And I have put this reference also in Nature Reviews. If you put about epigenetics, those things will come. So mostly environment or diet or lifestyle that affects epigenetic changes. And one more point I want to make, that if you are studying genome, then from any cell of your body, you isolate a cell and genome sequence will be the same. So one time only you have to sequence genome. But epigenome changes with organs or cell. So that is there. Another thing, in twins, when studies were done for different diseases, then it was found that in diseases like cancer, mostly epigenetic regulation is dominating, while in mental disorders or neurological development, it is more genetic. So exercise or diet, they are always good for you. And initially, when gametes are there, or zygote or embryonic stage, at that time you don't see during development any kind of mutation or SNP or any other thing, which are by environment. Genetics is different, but epigenetics starts developing by methylation or histone modification. So mostly initially it is environmental or maternal factor, but gradually it is mostly diet and lifestyle. And at different stages, we can collect some biofluid which can be used to detect epigenetic biomarker, which we can use, whether urine, stool, blood, or skin. And in this case, I have shown how environmental factor or lifestyle they affect. Suppose some pregnant mother is smoking, then not only her life is means they become sensitive or responsive to cancer, but that infant is also affected, and stem cells in the infant are also affected. In this way, 3 generations will be affected. And one example is here that aldehyde and nitric oxide, they present in cigarette smoke, and they induce phosphorylation of histones. So that's how it has been identified. And this is, for example, one paper is there where maternal smoking, that affected DNA methylation in these children. And there, the way like genome-wide association studies, when they are done, then for all chromosome in different loci, certain SNPs are seen that whether they are there or not are associated with disease or not. Similarly, epigenome-wide association studies were done in cohort where disease was not developed, and those people were followed, and their methylation, histone, or chromatin compactation, all those things were seen, whether those are done or not. So this study was done that. Another example was that maternal anxiety also affected methylation of this gene. So those kinds of papers are coming. And vaping can damage vital immune system, and immune system indirectly affected by epigenetics. To understand epigenetics more or do EVA's kind of studies, we need healthy population which we can follow for a long time, and we need such biospecimen which we can collect easily, like exfoliated cell, skin, urine, or blood, or we need long time follow-up so that in the same person we can see changes and individual variation is less. Although these are expensive, but this is the way we go, and these are called cohort studies. In epigenetic biomarkers, if we want to use for disease detection, diagnosis, or follow-up of treatment, then what we want that since these are environmentally induced and tissue specific, so exogenous risk factor and endogenous risk factor, all those in cohorts, they are measured. And what we do in our program, as I mentioned initially, that our program is epidemiology and genomics. So in epidemiology, we collect biospecimen and person-related information from different racial and ethnic groups. And these are the countries from which we have collected samples. More than 2.3 million subjects have been enrolled. Either we do cohort studies or case control studies. So those kinds of studies we do. These are the names of some cohorts like nurses health studies, physicians health study, multi-ethnic cohorts. So 62 cohorts we support in our program. And what is the criteria for membership? That more than 10,000 people should be in some cohort. And in that we study polymorphism, genetic susceptibility, or gene-gene interaction, or gene-environment interaction, and now epigenetic markers also. At NIH level also, NIH OD started one program. It's called Environmental Influences on Child Health Outcome, or ECHO. This is a congressionally mandated program, about 160 million per year. And what we want in that idea is that early time exposure is so sensitive to the at the prenatal, natal, or postnatal or infant that may develop disease. So along with cancer, what they are following, asthma, neurological development, prediabetes, and obesity. Those are also being followed in 50,000 pregnant women. Those have been enrolled, and some infants are born, they will be followed for 7 or 10 years. And this is an extension of National Children Study. And in that, epigenetics mark, more metabolomic marks, and all those will be collected along with environmental influence, social economic status, and lifestyle factors and others. And this is the organization, how we have made the coordination center or data analysis center. So at what time we are collecting the information on biospecimen? Preconception stage, prenatal, labor, through discharge up to 1 month, 1 month through 11 month, early childhood 12 month to 59 month, and then so on. So in the same infant or baby or child, we are going to collect the sample and see what kind of marks are coming. And later on, some will develop the disease, some will not. So wherever disease is developed, then we can go back to those samples which are collected in other people to identify disease early. So that is in utero and early in life and other transition period that we are covering. And these are the biospecimen: placenta, cord blood, nail, hair, saliva, urine. By doing NMR and MS, even from nail or hair, it can be identified what kind of exposure in the environment they had or parent had. So those kinds of things. In behavior or emotions also, epigenetics regulation has been reported. And this paper came about maternal anxiety, as I mentioned, in IGF-2 in histone. And epigenome-wide association studies have been done where they showed that aggressive behavior is reflected in terms of epigenetic marks. So it is not like other kinds of things which we see that fatal disease are there, because many things, as you understand, that by stress and psychological pressure and other things happen. So in that also epigenetic regulation is there. And this also I have written with one fellow about epigenetic regulation in biopsychosocial pathways. Now, one term we use in our epidemiology, it's called social epigenomics. The thing we collect that many populations you will find in different ethnic group that they live in such a place, even healthy food are not available, along with their social economic status and occupation and others. Generally, genetic marks come much later, but epigenetic marks come first. So that we are following in this population, and that we call social epigenomics. And one article I wrote, and that was that genome-wide association studies, or GWAS and EWAS, should be studied together. Then we can conclude better. In environment, how epigenome is affected and what are the agents which have been identified. Arsenic, benzene, cadmium, chromium, nickel, all these either affect genetic or epigenetic methylation or chromatin. So that have been identified. We have another institute like NCI, we have NIEHS, National Institute of Environmental Health. So we work with them. So one program I wrote for environmental influences on epigenetics. Cross-generation effects also has been studied in some cases, like alcohol dependence is regulated by methylation. And toxicogenomics, like whenever drug is developed, that does not work in all people the same. So genetic background affect which will be which person will give response to treatment or not. Similarly, we are promoting that epigenomic background also will tell whether some drug will work or not. That we call toxico-epigenomics. So whenever there is a environmental insult or other things are there, then first thing will change our transcription factor or cell cycle regulator. Then chromatin changes like promoter and methylation and histone. And it is after a long time, then only genetic things come. And this will take about 2 minutes, 5 minutes, or 10 minutes. These things take 8 hours to 12 hours. Then metabolomic changes 24 hours, but it takes few months by the time you see genetic changes. So then also we emphasize that if we understand early stages first, then it will be useful. In genetics, different modifications like translocation, deletion, amplification, indels, etc. have been identified. So in epigenetics, what we say that between genotype and phenotype, these two stages are also there. Epigenotype comes first, and metabolomics or metabotype comes second. Those epigenetic modifiers which are there, we wanted to see how much genetic and epigenetic interaction is there. So in those stages or genes where epigenetic regulation has been studied, those are shown here. So in some cases that has been done. Generally, when disease developed, then general global hypomethylation is there because of line and sign sequences, but in the promoter specific hypermethylation will be there, and histone alteration also increases with cancer development. And here line 1 methylation has been correlated with mortality of person in prostate cancer. Now DNA methylation also has 3 stages, like it can be abnormal increase or decrease or no changes. When increase is there, like in tumor suppressor genes, either methylation of both alleles or one allele methylated, another mutated, or hypomethylation of allele and mitotic recombination in others. When decrease is there, the way it happens in viruses, especially retroviruses, when gene therapy came, then recombinant viruses, many times they did not work. The reason was that their LTR, which is equivalent to promoter, that get hypermethylated, and that's why that was inactivated. So in retroviruses or lentiviruses, that has been studied. When there is no change, then that is by chemically induced changes or poor repair of methylated cytosine or deamination. So different states are there of methylation, but now technologies are such that even for full pathway, you can identify which genes will get regulated by methylation. And in those experiments, what you keep in mind, that methylation content or methylation level or pattern or level profile or pattern profile you do, and that we do to reduce false negative or false positive results. Steve Bellin and Johns Hopkins, he has identified different proteins and their addition of sequence to regulate heterochromatin and euchromatin. And then example is here like PRC, one protein, if this trimethylation you see after lysine 27, then it will be involved in differentiation. Similarly, if S3 followed by lysine monomethylation, and next amino acid is serine or serine, then upon phosphorylation, this change will be permanent. So those kinds of detailed analysis has been done. Basic idea is that histone modification, when they happen, they contribute to cancer development. MicroRNAs, 10 years ago, they were reported only in plants or Drosophila, but then in human they were reported. And now we know that for different cancers also, microRNAs are there. They are only 20 to 25 nucleotides long, and they have a stable structure. Because of that, we can use either genes of microRNA coding genes where there is any polymorphism, or just microRNA profiling. A set of microRNA will indicate quite early whether cancer is developed or not. And here it is shown, sometimes microRNA will activate some genes or sometimes inactive. So they also play a role simultaneously. Now, one more concept I want to put, which is for extracellular vesicles. In the body, different organs, they will release small vesicles. These are double membrane protein, but they will contain some part of DNA or RNA or protein or microRNA. These are easily accessible, and those can also be used as a biospecimen to see whether epigenetic modification or genetic modifications are there or not. So this article I did for review purposes. This picture shows histone structure. These are different modifications in histone, S2A, S2B, etc. So at NIH, we initiated one program, it's called Epigenome Roadmap, in which epigenome profile of healthy people was done, and modification of these histones. So against these modifications, monoclonal antibodies were raised, and free of charge we distributed to investigators so that they can study which modification is contributing to disease development. In histone, they will explain where is the active promoter or inactive promoter or enhancers, different kind. So in that, this helped to understand that. H1 protein that works just like a linker. This is another representation of histone. In histone now, technologies are such that for different histone at different time, we can follow what kind of modifications are there. And these are some of the examples where histone modifications are shown, monodimethylated or trimethylated or histone, and whether they activate or inactivate gene. And now some technologies have come where in different samples, single cell epigenome also you can do. For basic studies, these kinds of approaches are needed. And now histone modifications can be used for diagnosis also. So that article I wrote for that purpose in different cancers. At different times, I have edited different books, how cancer epigenetics is developed and how much those host-specific markers or environmental marker they work, how we can prevent or biomarker, or in precision medicine, how we can use that. One time I was interviewed by Nature and they asked me, why do you think epigenetics is more important than genetics? Then I mentioned that if you study genetic changes, you can use at the most for diagnosis or follow-up, but epigenetic changes by drugs or chemicals, you can reverse those changes. So then role comes in cancer control or prevention. Another time also they interviewed, and that time I mentioned that different companies are now developing, and FDA has approved also some drugs which are called epigenetic drugs, so they are very effective. Genetic changes you cannot do, but epigenetic changes you can modify. Sometimes exfoliated cells from tumor, time to time they are disposed. Those can be collected at different stages of cancer development, and then you can see that what kind of biomarkers are expressed. And these are group of genes which are regulated by epigenetics and in different cancers, and a number of articles or review articles I have done. So if you want to know more about that, you can read those. These are those proteins or the epigenetic modifiers, and these are the genes which are affected. Those are summarized there. The reason of summarizing, as I mentioned, epigenetic modification can be reversed. So whenever we see some changes, then different pharmaceutical companies are making intervention agents or therapeutic targets against those. These are some examples. These are the names of those drugs which are being developed in different phases. And as it happens while developing the drug, that sometimes side reactions will be there. But still, these are the names of those drugs which are in the pipeline for different epigenetic modifier. And whether it is Johnson & Johnson or Schering-Plough or Merck or other big pharmaceutical company, AstraZeneca, every company has a program, big program in epigenetic drugs or epigenetic modifiers. And they cover different classes of chemicals like short chain fatty acids, aliphatic compounds, or cyclic tetrapeptide or benzamide. And these are for cell cycle arrest or differentiation, migration, or apoptosis. So they are taking different stages, and during that time, what kind of modifications are there, and then they are developing the targets. I will give you two examples. Now, later on, what was observed that either single agent can be used for treatment of cancer or combination. So in some such cases where other reagents or other regular anti-cancer drugs were not working, and in this case 55 people were such, and valproic acid and azacitidine for methylation, this is for acetone, when in combination that was used, and then in day 1, 10 and 28, like methylation sequencing or histone sequences or microRNA you can do in high throughput. When it was used and followed, then those could be treated. Similarly, in another example, retinoic acid in myeloid leukemia or myelodysplastic syndrome, 5-azacitidine or trans-retinoic acid, they were used. In this case also more than 50 patients were treated. So sometimes these agents work as radiosensitizers. It means regular anti-cancer drug is not working, but if you pre-treat with these epigenetic drugs, then they start giving response. And these drugs have done in solid tumors as well as lymphoma, leukemia, breast cancer, glioblastoma, myeloblastoma. So here I have shown for histone inhibitors, those drugs, and these are the methylation inhibitors. So in different categories, those have been done. So these are not only theoretically that has been explained about epigenetic inhibitors, but they are being used, and different companies are making these drugs. So whenever nowadays treatment is done, then genetic aspect as well as epigenetic aspects and both kinds of inhibitors are being used. Initially, FDA approved 4 inhibitors, 2 for methylation pathway, 2 for histone pathway, and these are the names. And whenever, as you know, FDA approves, it means that can be used in patients. But now 4 compounds or 4 drugs have been approved which are as DEC inhibitor, histone deacetylation inhibitors, or 2 for methylation inhibitors. Those have been approved. And if you want to study more about that, like Nature Reviews gave this article, when you type cancer.gov and type clinical trials and epigenetic drugs, then also you can see about that. So they will tell you about which therapy is there, what is the combination of drug, how patients were selected, and how many did give response. So these are some examples of different cancers where clinical trials are being done for new drugs also. Now, pharmaceutical participation also in combination drugs, especially like decitabine or EZA, these are the epigenetic drugs and these are the regular drugs. So that has been done. And even like here in case of leukemia, AML, subtyping is also done based on, for different subtypes, different combination is working. Now the way they work is which agent should bring first and how much will be the dose, that is there. So I have written for colorectal cancer about epigenetic therapy. And this is one example of one review article I will recommend you to read. Combination epigenetic therapy in non-small lung cancer, or this one is for hematological malignancies. So few examples I will give you. Like in colorectal cancer, those genes have been shown which is different stages are regulated by epigenetics. And in a large number of people, when methylation profiling, along with that SIMP phenotype, which is called CpG Ireland methylator phenotype, or KRAS mutation and BRAF mutation, and microsatellite instability high or low, all in combination was done. Because in biomarker field, it is understood that instead of using one biomarker, if we use more than biomarker, then sensitivity and specificity is more, and you can make a better decision that who is responsive to treatment and who has the disease. So for diagnosis, prognosis, or detection, it is there. So by this, it was shown that if you combine methylation profiling with MSI or mutation, then you can select a combination of genes to diagnose colorectal cancer. Similarly, methylation profiling could distinguish healthy from AML, ALL mixture or ALL only. In other example, in lung cancer, combination of protein markers with methylation markers was done, and these are the genes which are regulated by epigenetics. And sputum was collected from that because it was non-invasively collected to identify differential methylation. Mesothelioma is also a type of lung cancer. In those people who were exposed to asbestos, in that also methylation profiling indicated that who is at high risk of developing mesothelioma. Another cancer, esophageal cancer, which is the early stage of esophageal cancer, Barrett esophagus. In that also, different genes were followed for methylation profiling. And as you see, wherever darker region is coming, and it is in the same person, this is also same person. So at different stages, methylation profile can be seen. And methylation profiling has been correlated with survival as well. In pancreatic cancer, what happens that ovarian cancer and pancreatic cancer are such that by the time you detect, then it is too late and survival is 5 years or 4 years or so. So what we do and why patient-related information is important, if somebody is diabetic, alcoholic, and they have KRAS mutation and P16 and P14 are methylated, then that person is at high risk of pancreatic cancer. And that has also been seen to distinguish different stages by chromatin states, where transcription is more, transcription is there, and chromatin status, compactation, and relaxation. In breast cancer, response to tamoxifen treatment was indicated by methylation of selected genes. And based on that, like you can do intraepithelial neoplasia and distinguish from in situ, and sometime over-treatment is there to distinguish which population or which person should be treated or not treated, to distinguish from aggressive to indolence, and that also methylation profiling help. In 10 to 15% in cancers, infectious agents are involved, and as you know, HPV 16 and 18 for cervical cancer, or HBV, HCV liver cancer, or Epstein-Barr virus for nasopharyngeal carcinoma, H. pylori for gastric cancer. So latency associated nuclear antigen, these are the genes which were associated with that. And what we wanted to do to utilize epigenetics, those biomarkers, so that we can identify asymptomatic people who are from internally they are being affected, and we can identify either in early genes or late genes how much methylation is there, which gene is methylated, and that indicated which stage of cervical cancer they are and where cervical cancer will develop. And that information we use for triage, where treatment should be done or treatment should not be done, and which genes are methylated more, and about invasive cancer versus in cervical cancer, many times HPV infection is there, but person will not develop cancer, to identify those also. And for different viruses also where cancer is associated, those have been studied and epigenetic regulation has been identified. This example is for liver cancer because hepatitis B and C, they are there. And in liver cancer, these are the genes which are regulated by methylation. In T cell methyltransferase, in lupus and cancer, that has been identified. So many genes which are regulated immunologically, there also it has been studied. In prostate cancer, PSA is the main biomarker, but whenever level is more than less than 4 nanogram per ml, then it is difficult to tell whether person will develop prostate cancer or not. In this case, GST-P1 was the one which can detect early, and Quest Diagnostics from Epigenomics company, they bought that, and that kit is now out, which is used to detect prostate cancer. In bladder cancer, exfoliated cells from urine, they are collected, and methylation of laminated gene, that was followed to detect that bladder cancer. Similarly, in some example, different tissues simultaneously you can tell, because in cancer what happens if it starts in one organ, then some peptides and proteins and other things will go in other organ, and second is cancer will start coming. So that way simultaneously you can study those. Now a little bit about prevention. It is understood that 30 to 40% cancer are preventable by changing lifestyle and diet. So there also we emphasize so much. And these are some of those food and their bioactive components which have anti-methylation or histone modification properties. So those also have been studied. And this article I wrote for Cancer Control and Prevention, Nutrition and Epigenetics. And in that also, we have emphasized that what are the food and their bioactive food components which have demethylating activities or deacetylation activities. In case of liver cancer, when only methyl donors were utilized without any added carcinogen, then such model was created which was similar to liver cancer. So in hepatocellular carcinoma, how you can prevent, that has been explained in this paper. And this figure shows some regular food like tomato, apple, soybean, turmeric, cinnamon. Some have histone modification properties and some have DNA modification, some have both. So many times it is advisable, like on diet they emphasize so much. Now gradually we are understanding that these food have those properties. So with these kinds of studies, what advantage we have or what kind of research we can pursue, that to identify these epigenetic marks, whether they can distinguish those races or ethnic groups which are at high risk of developing cancer, or to indicate whether case control studies or cohort studies we should do for those studies, or how much genetics and epigenetics correlated, or whether microRNAs or non-coding RNA can distinguish race and ethnicity specific responsiveness, or EVAS studies, how much useful it is, whether we can predict recurrence or secondary cancer. Or nowadays, that is also difficult to harmonize data like from different population when we collect to develop bioinformatic technologies, which is the window of susceptibility. Or since these inhibitors might affect normal cells, how to avoid that? What is the role of non-histone proteins? Cancer stem cells, in that what is the epigenetic regulation? So those kinds of challenges and research opportunities are us. And now a little bit in last 5-7 minutes, I will tell at NIH and NCI how we are addressing those challenges. At NIH, when Zerouni came as NIH director in 2004 or 5, he initiated one program and proposed that all institutes of NIH, they will contribute 1 to 1.2% of their budget, and research will be done on such topic which affects many diseases and common mechanisms are there. So as a first step, microbiome and epigenetics were selected. And the idea was that epigenetic was selected because by that time we knew that different kind of, whether it is cancer, neurological development, cardiovascular disease, diabetes, everywhere some indication were there that they are being regulated epigenetically. So this program provided that such kind of resources so that these studies can be done, and healthy people were enrolled, which were pathologically, they did not have any disease. And then from those, okay, let me show you one slide first. From those, what was done, that these are different organs. So 111 kinds of cells were collected in different organs, and methylation profiling and histone profiling and microRNA profiling was done in that. And these were called roadmap program. And after that, other topics were also came. And first one was called epigenomics. Later on, in which I am involved, I represent NCI. One is called molecular transducers of physical activity, in which epigenomic profiling along with metabolomic profiling, genomic profiling, how they are affecting physical activity, that is there, or metabolomics as such, how they affect cancer and others. So this is the program, and annual budget is about $500 million. And for epigenomic program, it was started from 2008 up to 15, and then 2 years it was extended for functional epigenomics. So that's how it was done. And I mentioned about monoclonal antibodies against histone modification, so those were done by this program. And in one issue of Nature Whole, the way genome map was done, similarly epigenome map was done, so that has been published there, and about 15 articles came around that time for different organ sites. This I showed you. And with that program, a number of high quality publications also came in Cell, Nature, or Science, or others. So if you put epigenome, then those things you can read. After that, one international symposium was also started. It's called IHEC, or International Human Epigenome Consortium. In that, 1,000 epigenomes are being constructed, and they will be the reference map for different stages. And they are working on age-related epigenetic changes as well. So first meeting we had in Paris, where these many countries participated, and different funding agencies, they put money. We wanted that whatever we are doing at NIH, it should not overlap, so I represent there in IHEC program. And then Affymetrix provided equipment to do the methylation analysis, and other Novartis also supported that. And at the time about this program, it was published in Nature and Science. And this is the website for IHEC. Another important thing is to understand basic science. Human and animal, both studies, both epigenome are being studied in IHEC program. Time to time, we write article also to show that in which area we are emphasizing or prioritizing. So this one we wrote for epigenetic research in cancer epidemiology, what are the trends and challenges. Similarly, I did this one for molecular profiling and companion diagnostics, how you can use epigenetic biomarkers. This one was about molecular transducers of physical activity, or we call it motor pack. And in that, tissue samples like human blood, muscle, adipose tissue, and animal model also, they will be done. And all these things like genes, protein, peptide, lipid, sugar, epigenomics, microRNA, metabolomics, all this will be done. So for 13 weeks, a group of people will do exercise for certain time, 30 minutes. And how much these molecules are changed, that will be identified, that how much it is essential and not. And these are different components and amount which is shown. This is a 6 to 7 years. This will continue. Just 3 years ago, we started. Few more programs are being done, like one is called Moonshot. Biden, Vice President, he started that after his son died. So dedicated about 300 million per year NCI got for 7 years. And idea was that whatever we are doing, we continue that, but can we develop drugs or treatment or diagnosis, whatever we are doing in 10 years, whether we can do in 5 years if additional funds are provided or not. So 2.5 years ago, this was started, that is going on. Similarly, one program was started, it's called All of Us, in which 1 million people are enrolled, and they are being followed, that how much, which disease they develop, and their social economic status and family history, that will be done. One is called Cancer Genomic Atlas, similarly Pre-Cancer Genomic Atlas. So these two programs are also going on. So that's how we are addressing those challenges. So now I will take any questions which you might have. Thank you.
Thank you. Any questions?
Yes.
So just in terms of a clinical workflow, can you just speak on what the challenges would be of doing like epigenetic profiling versus genetic profiling of a patient's cancer, and how those are different, how feasible they are?
So when genetic background was done, and that was done initially by mutation analysis, and later on mutation covered only a small part of genome, then they thought we should use SNP, or small nucleotide polymorphism. And genome-wide association studies, or GWAS, were done. And that we spent millions of dollars and 8 or 7 years at time, and for different cancers and different diseases. But most of the SNPs and other things, they were coming outside the genome. So generally, whenever target drugs are produced, they are in the coding region. They were not there. And still many things were not covered. And as I indicated initially, that when monozygotic twins were followed and discordance was studied, then it was found that epigenetic regulation is so much prevalence. And by genetic studies, it was found that only 8 to 10%, especially in cancer, is hereditary. Remaining is environment and lifestyle. So then epigenetics came, and second point, genetic changes you can just follow how the person is doing, whether we can use that mutation for pharmacogenomic purposes or diagnosis or development. But epigenetic drugs, when you use, you can reverse that. So that was the advantage, and in combination therapy it is working. Now in terms of challenges, that epigenetics, these regulations are different for different organs or cell. So how much it will work, for which organ it will work. So for that, they are working on different histone modifications or microRNA, whether they can make it organ specific or not. Most of the success is in colon cancer and prostate cancer, but in others it is ongoing. So combination therapy is promising, and as I indicated, so many trials are being done, and 6 drugs have been approved also.
And then if I have one more question.
Yes.
If you had a gene that was commonly hypermethylated, for example, is there any way to target just that gene, or are these pharmacological agents more general, and you hope that that gene's more affected in the cancer cells?
So whenever patients are treated, then not only these drugs, but the family history, what is the lifestyle, what was exposure, whether he has another kind of disease also or not, all those things are also kept in mind. And then in combination also, which agent should come first, which agent should come after that, and how much dose to start with. That way it is done. And most of the drugs, when they were identified or discovered, then first they were used for those patients where no agents or no anti-cancer agent was working. When they saw some response, then they started giving to others. And then they found, especially with PARP inhibitors, these STAC inhibitors work very, very nice. But as it happened, that it is difficult to get drugs, and everybody does not respond the same way. So we emphasize the way people do pharmacogenomics. About 10 or 12 genes are such, where genetic background will tell whether you will respond to drug or not. Except BRCA1 and few others, we don't have many. So now we are working on developing epigenomic profiling, whether that can help us to identify or stratification of patients who will be treated.
Thank you.
Okay, I think we'll be moving on. Thank you, Mukesh.
Thanks.
Our next speaker is Peter Choyke. He's the director of the molecular imaging program. So he got his MD from Jefferson Medical College. Then he did a diagnostic radiology residency at Yale New Haven Hospital. He did an imaging fellowship at the University of Pennsylvania. He joined the faculty of Georgetown University. And then he came to NIH. So he's going to talk to us today about a new topic, near-infrared photoimmunotherapy, a new light-based treatment for cancer.
Thank you. Yeah, I'm shaking it up, Terry. Very excited about this topic, a new therapy that was developed in our lab. So no financial conflicts of interest. The patents that I'm going to talk about have been licensed to a company that's now called Rakuten Medical. And I want to start by acknowledging my colleague, Dr. Hisataka Kobayashi, or HK, as we call him. So light therapy is nothing new. Of course, we use light to burn tissue, for instance, laser ablation. It's commonly used in plastic surgery to ablate discolored lesions. And it requires very expert control of the laser beam so it doesn't burn inappropriately. There's another kind of therapy called photodynamic therapy, in which a compound called a photoporphyrin is injected. It circulates and preferentially takes up in tumors more than normal tissue. However, it also takes up in normal tissue. And so there's therefore a very narrow therapeutic window, and there can be huge problems with this. So with photodynamic therapy, you inject this photosensitizer. It supposedly concentrates in the tumor. You then apply light to it, and in theory, the tumor is selectively destroyed. However, there are very bad side effects associated with this, which limit its efficacy. And I'll show you a picture in just a second. It also kills by a process called apoptosis, which is not particularly immunogenic. And the patient remains photosensitive for 2 to 8 weeks, so they can't go out in sunlight. So all in all, it's not a very appealing therapy. So this is actually Meyer Betts, who injected himself with hematoporphyrin and then exposed himself to 10 minutes of indirect or direct sunlight. And you can see he swells up. He was probably miserable for several weeks after this. This is pretty much what happens to the organs that are treated with photodynamic therapy. So we used to do it at the clinical center. There were very elaborate setups with all these laser catheters that were applied during surgery, et cetera. And there are actually some pictures of Bill Sindelar from the surgery branch in the mid-1980s doing photodynamic therapy in the thorax and in preclinical studies in dogs. So, and then Harvey Pass also did this kind of work. But clearly, there's much to be improved in photodynamic therapy. So if we borrow off the idea of fluorescence, we can use a dye that we excite with light, and that will emit light at a slightly different wavelength. So you might come in at this color, and it emits at a slightly different color. The whole concept is that there's an excitation by the light that puts the electrons in a higher energy state, and then the electrons return back to their basal state, and in that case release light. But they can also do other things when they come back. They can release heat, or they can induce chemical reactions. And this latter is what we're going to be talking about in photodynamic therapy. So we are an imaging lab, and our initial idea was to develop targeted imaging fluorescent dyes. So we took antibodies, in particular trastuzumab or Herceptin, and basically bought out the catalog of photosensitizers that we could find in the near-infrared range, near-infrared being ideal because it travels through tissue further. And so we would set up different amounts of this dye conjugate with the photosensitizer and apply light to these various wells. And at the time, this was about 2010, Hisataka is pictured here with Michelle Longmire, who's since gone on to become a dermatologist, but Mikako Ogawa, who's a scientist in Japan. And they did the original work with us. And what we found was that you had dyes that were pretty bright. They looked pretty good. And then ones that were moderately bright, and then not so bright. And then there was this one, these cells that were dead when you applied the light. So the light killed the cells. First we thought, that's not such a great thing. Sounds toxic. But we realized it's cancer cells that are being killed. That's a good thing. So maybe we're onto something. So this very particular dye, IR700, became very interesting to us. So this is the chemical structure of this dye. It's a phthalocyanine dye, and it has two side chains here. And it turns out when light hits this, these side chains are jettisoned. And this molecule, which is ordinarily very hydrophilic, becomes very hydrophobic very rapidly. And we believe that that's the reason why it's so effective. So when we apply light to this conjugate, especially when the antibody is at the cell membrane, it causes damage to the cell membrane, leading to cancer cell death. So this is a video of what happens to the cell. It's slightly speeded up, but it's not terribly speeded up. So there's the exciting light, and then you see the cells bud, and you'll see them burst. And the dye that's inside the cells will slowly fade out as it disperses into the extracellular space. So it's a very unique kind of cell killing that causes a near instant cell death. So it takes about 30 seconds for this to happen. And over the course of 1 to 2 minutes, it's completed. So it's much more rapid than most of the other kinds of cell deaths. But very importantly, it doesn't denature the contents of the cells. It doesn't damage the contents of the cells in any way. It simply releases them into the extracellular space. It's very specific. So before I get into that, here we see it at 37 degrees. You can see the swelling of the cells. So it works at room temperature, but it works at 4 degrees centigrade, which tells us that it's a process that is very much on the surface of cells, because if it's at low temperature, the conjugate can't get inside the cell. So it's a membrane effect. And then we have cells of two different types here. One is HER2 positive, one is HER2 negative, right next to each other. And the light is applied, and only the HER2 positive cells are affected. So the immediate adjacent cells don't seem to have any effect on it. So it's super selective for the cancer cell of interest and a transmembrane effect. So what we think happens is that the antibody with the IR700 binds to its cognate receptor in the cell membrane. And after the near-infrared irradiation, there's damage to the cell membrane, making microperforations in the membrane, allowing water to flow into it, causing cell expansion and cell death. And so the contents of the cell can then emerge. And here's another example of a cell being treated with this, and you can see how the cell grossly expands and bursts within just a few seconds of the exposure. This is a movie that I created at great expense to my branch. But it's not even playing. How bad is that? Oh, wait, it's got to play. It's too expensive not to play. So hopefully it'll progress. There we go. So the sound is not that important. I can talk over it. But you can see the antibody, and we attach these IR700 molecules, and then we inject it into the patient, and it travels around the body and finds the receptors it's interested in and accumulates there, escapes from the blood vessels, and binds to the cells at these receptors. And then, and this is the best part, the light is applied, and the damage to the membrane starts to take place, and the cells start to swell, and eventually there we go. So it bursts and it releases these cellular contents. And we have good reason to believe that's what happens. We've been able to see these small filopodia among cancer cells disappear very rapidly after the application of light. You can just see them getting trimmed away. There's a control where the filopodia don't get damaged at all. And this is the haircut slide with this very furry looking cell, and then you apply the PIT and all the little filopodia disappear right away. Whereas the control, nothing really happens after the exposure to light. So we put this we were very excited by this finding, and we did what everybody would do. They put it into a mouse model. And so we implant 2 tumors in this mouse. One is a control that doesn't receive near-infrared light, and this tumor will receive the near-infrared light after the administration of this conjugate, panitumumab IR700. And this is the white light image, so you can see both of the tumors are fluorescing a little bit. And then we apply the therapy, and you can see the diminution in the signal and the decrease in the size of the tumor compared to controls. And to be honest, I mean, this was a good result, but it wasn't super spectacular because you can see the regrowth of the tumor. And one thing you could do is do it again, and that would knock it down again, and you got an effect like that. And there was prolongation of survival and things like that. But we were excited about it, but we didn't think it was going to be the answer to all of our dreams. But there was a very thoughtful company that had initially named Esperion, now purchased by Rakuten, that was willing to take a chance on it and license the patent. They went on to do toxicity studies using cetuximab, which is a EGFR antibody, IR700 conjugate, and they tried it in non-human primates, NHPs, and there was no apparent toxicity with it. There was no systemic toxicity, so they funded a phase 1 dosing study in inoperable head and neck cancers. So just briefly about head and neck cancers, it's a very common cancer. 2/3, this is not just the United States, this number is worldwide. 2/3 occur in developing countries, but in the United States, it still accounts for 12,000 deaths. When we talk about head and neck cancer, the various organs around the mouth, the back of the mouth, the glottis, the larynx, can be affected with head and neck cancer. And it's a very debilitating tumor because our breathing, our swallowing, our tasting, all of these senses are in this area. And the treatments that we give patients often result in severe damage to one or more of those functions. And the current treatment is chemoradiation followed by surgery. And then if the tumor recurs, combination chemotherapies, and you can see a pretty poor response rate, 10 to 36% in that setting. People receive immunotherapy and antibody therapy. reirradiation can cause very significant toxicity. Our friend photodynamic therapy has been used, but there can be severe side effects, carotid rupture, fistulas, etc. So this is a space that needs improvement. You know, it's not very good situation right now. And so that was the space that we entered in. And in the phase 1 study, as you know, you're trying to find a dose-limiting toxicity. So you start at a low dose, you go to higher and higher doses, and you see where you start to see toxicity. So because this is a 2-part therapy, it has both the conjugate and the light. We thought we had to increase both of them. And so a multi-center trial was begun. And by the way, you know, not all of the light needs to be exposed to the surface. We could insert fiber optic probes into the tumor to deliver light into the tumor if it was not amenable to surface illumination. And this is just, this was the preclinical setup that we had, just a simple box with LED lights. But when we go into the clinic, we're using these fiber optic probes that can be inserted into the tumor. And we were very surprised by the results of the first 4 patients. Remember, we're just trying to establish toxicity here, not necessarily treat the patient. But the first patient had a 70% reduction in their tumor size. The second patient was actually a complete response with 100% tumor death. The third patient had a greater than 70%, and the fourth patient, all at the lowest doses, had a complete response. So we knew, you know, this is sort of an unusual result in a phase 1 trial because the results are quite good at the lowest doses, but they're also much, much better than we saw in the mice. And so what's the difference between a nude mouse and a human being? The immune system. There's an intact immune system in a human, but the mice we use have the T cells knocked out so they don't reject the tumors. So here's some pictures from some of the treatments. This was a tumor in the mouth. You can see this is the uvula. And then right afterwards, there's a whitish discoloration to the tumor. And eventually, after a few days, the tumor sloughed off. The patient could actually watch it in the mirror, and it was one of the complete responses. And so the people participating in this trial were very excited about it. One of them was Rush Medical Center, and they did some Eureka alerts and things like that, and talked about how almost immediately you can see the tumor die, turn white, and it's destroying cancer cells, causing almost no damage to surrounding cells. And that's really the remarkable part of this, is that it doesn't really damage other things except the tumor. So a lot of testimonials. This is a really pretty, so we went on to a phase 2 study with some really aggressive cancers. So this is a man who presented with recurrent head and neck cancer. You can see all these lumps in his side of his mouth. And then we place the fiber optics into the tumor, as you can see here. And 1 month later, there's dramatic shrinkage of the lesion. And at 3 months, yes, there are still some fistulas and things like that, but there's no tumor at the site. And in fact, his hair, the beard, is starting to regrow hair at that point. So the results of this phase 2 trial were that, again, minimal side effects, no photosensitivity that we see with photodynamic therapy. There was a nearly 100% response rate and a 57% durable response rate. And so we're going on to conduct a trial that's going to open soon in a phase 3 trial at NCI very shortly. So the response rates in the phase 2 were quite good, nearly 14% complete response rate, 31% partial response rate, and 38% stable disease. A few patients did go on to progressive disease, but overall, this is a very impressive response rate in this heavily pretreated population. And more importantly, the patients lived longer. And so we can consider this as a potential third-line therapy for cancer at this point. It's not approved yet, of course. It's still in phase 3 testing. So we go back to this slide that I showed before in mice, where we had these regrowth curves that I mentioned were not all that impressive. Yet the patients seemed to have a very dramatic response. So we sort of mentioned that the immune system might have something to do with it. And here's another patient with multiple nodules, and you can see 1 week after they've all turned color, and 1 month later they've really sort of gone away. And 2 months he seems to have a normal neck, which is pretty remarkable. So this is the difference between a xenograft and a syngeneic model. So if we take a human with a tumor, we take the tumor out and then passage it so we can grow it into something called a cell line. We can put it into a mouse with no immune system because the mouse won't reject it. But if we want an intact immune system, we have to stay in the same species. That is, take a tumor from a mouse, passage it, and then put it back in the same kind of mouse. And then it will grow, usually more slowly, because now it's being attacked by the mice, the recipient mouse's immune system. And that's kind of, that's the situation that we find ourselves in here. Now, there are many kinds of immunotherapy. You can get, and I don't want to dwell on this slide too much, except to say that the immune system is very complex. But our idea behind what goes on in cancer immunity is that there will be T cells that traffic to tumors. They'll get through the endothelium, they'll infiltrate into the tumor space. The T cells will recognize cancer cells by immune recognition. They can even kill the cancer cells. And when they kill the cancer cells, they release antigens, which are picked up by dendritic cells, and they prime additional T cells to effectuate an immune response. In the case of what we're doing, we think what's going on is that because the cell explodes more or less upon the application of near-infrared light, there is the release of multiple antigens into the environment, as well as various damage-associated membrane proteins, such as CRT, ATP, HMGB1, that have a direct effect on the dendritic cells. And they receive these antigens and mature from immature to mature, and then present the antigens to T cells, thus educating them. And then the T cells release granzyme and interferon gamma to kill the cancer cells. Now, that's a great story. There's some problems with it. One is that there are a lot of inhibitory T cells in the environment. For instance, T regulatory cells that have a negative effect on these T cells, and that'll degrade the immune response. And there are other suppressor cells. So we believe that we're getting quite a powerful immune response. But what if you could also take out at the same time these T regulatory cells? Might you also be able to have a very profound effect? So we started to do those kind of experiments in which we did in mice near infrared PIT with local knockdown of T regulatory cells using a anti-CD25 IR700 conjugate. And again, we implant 2 tumors. This time, we're going to be looking with luciferase bioluminescence imaging. So there are 2 tumors, and we treat this tumor. And within a day or so, that tumor disappears, having only taken out the T reg cells. But look what happens to the other tumor, which with direct tumor PIT simply went on to grow. In this case, both tumors regress, and actually pretty rapidly within 3 or 4 days. So that was sort of an interesting result, and we repeated it in other models where we implanted 2 different kinds of tumors diagonally. So these 2 tumors are the same, and these 2 tumors are the same, but this is not the same as that. And then applied PIT just to this tumor, and a day later, not only this tumor, but its opposite, which is of the same type, is gone, whereas the other 2 tumors are still there, albeit a little knocked down, but still very viable. So we believe that in the pretreatment state, the tumor cells are more or less shielded by T regulatory cells from CD8 and NK cells in the tumor microenvironment. After the near-infrared light irradiation, however, with the demise of the T regulatory cells, these T cells can now have direct access to the tumor cells. And eventually, because they expand so quickly in the presence of cytokines, they spread to other parts of the body where they look for tumors of the same type and kill them. So it's really harnessing the immune system to its maximal potential. So, you know, T cells, as I mentioned, can cause all kinds of effects. They can be activated by antigen-presenting cells, but they can be downregulated by regulatory T cells, myeloid-derived cells, and a variety of other cells. So it's a balancing act. Now, you may have noticed that I specifically refer to the antibody we used as an FAB'2, which is not really the same as an antibody. If you know your antibody lingo, it's a fragment of an antibody. And so why did we do that? Well, if we used the full antibody, we would have we could have eliminated the Tregs, but the antibody would still be around, and activated T cells start to express the same antibody. So we would, in essence, defeat our purpose by having the antibody that binds to activated T cells as well. By using an FAB', we can eliminate the fragment faster, and also eliminate any interference with IL-2, which is what actually which is what CD25 binds to. It's called the IL-2 receptor. So to proliferate T cells, you need IL-2, and we don't want to block the IL-2 on the CD8 cells, the immune cells. So this is a trick that we use to get the killing we want of the Tregs without damaging the CD8s. So we've explained that. So what about if you combine the treatment of the Tregs with treatment of the cancer? That would seem like a logical next step. So we did that in a syngeneic model. So now we've switched from nude mice to syngeneic models because we need an intact immune system. And we have various controls. And this is the combined near-infrared PIT survival curve. And there's prolonged survival in those treated mice. There is some growth in some of them, but this is only a single treatment with both of those targets. And we can get really prolonged survival in these mice with very good control of the tumor growth using that. And in this case, in this model, we don't apply light to 3 of the lesions, but 1 of them is exposed, and you can. So in the control, nothing happens. All the tumors grow, and we have to sacrifice the animal. But in the treated mice, you can see that after a few days, you can get a complete response in this mouse, even though you've only treated 1 of the 4 tumors. And interestingly, if you try to re-inject these same cancer cells into this mouse, you can't do it. It will reject the tumor cells. because it's developed an immune response against the cancer cells that prevent you from re-injecting. So in essence, a tumor vaccine. So we're on our way to developing exactly this kind of combined treatment where we apply an antibody that will bind to the tumor that stimulates the immune, the antibody presenting the antigen presenting cells, resulting in maturation of the dendritic cells and activation of T cells, but at the same time taking out Treg cells so that these effects are not blocked. And we get a very powerful direct tumor treatment with vaccine response later on. So a multi-step procedure. So there are some other interesting aspects to this therapy, and that is that when we administer it, we see a dramatic increase in permeability in the vessels to the treated lesion. So, for instance, we've treated this lesion with PIT and then put in a quantum dot, which is quite a big and very bright fluorescent construct. And you can see the side that that received no therapy doesn't really enhance at all. But the one that did receive therapy is avidly enhancing. In fact, when you measure this difference between here and here and here and here, it's 24-fold increased permeability in the treated tumor. So that will enable us to deliver very large macromolecules to the treated tumor, preferentially to other targets, which means we can dose reduce. So photoimmunotherapy is a disruptive technology. It certainly produces dramatic results. It's already in head and neck cancers, and there are any number of other targets that it could be directed to. So we could go on to other tumor targets like prostate-specific membrane antigen for prostate cancer, or EGFR and FGF for bladder cancer. And your imagination is the only thing that limits the combinations that we could do. So it's another kind of interesting question is, who is this addressed to? Is this for surgeons, or is this for radiologists? And the answer is that it probably can be done by a lot of different doctors. Initially, we're going to rely on surgeons in operating rooms, but over time, I think we're going to be able to shift to outpatient delivery and basically a strong arm and a fiber optic catheter. And we'll be able to do quite a number of treatments. So this is my acknowledgement slide with a lot of people in my own lab and people at NIH, as well as collaborating institutions that have helped us get to this point. And that's Hisataka and his current team of postdocs. So with that, thank you. Any questions?
Perhaps you can elaborate on the phototherapy.
You mean how it works, or?
Just the methods that are used and how long you have to wait after the antibody administration.
So you administer it 24 hours before the light treatment. It's usually uneventful, that administration. It's sort of equivalent to receiving a single dose of the antibody for therapy, but it's just a single dose. And 24 hours later, the patient is brought to the operating room. That's where we're set up to do it. And if the patient needs fiber optic placement, typically under ultrasound or CT guidance prior to coming to the operating room, the fibers are put into the tumor, and they are then hooked up to the laser system. The appropriate wattage is dialed in, and then the tumor is treated while the patient is under general anesthesia. that's because if you don't do general anesthesia, there's some pain associated with the procedure. But the recovery is very rapid. Within 2 or 3 hours, the patient is feeling better from the anesthesia. And really, many patients are discharged home at that point. But of course, at NIH, we admit them and do all kinds of tests. But many are feeling very, very well. We've had very dramatic stories where people come in and are not able to swallow because of the tumor, and they have a feeding tube or a gastric tube. And after the therapy, they're able to swallow and actually drink for the first time in months. Very gratifying. Of course, the complete response takes 1 or 2 months for the tissue to heal and to get full recovery. But the patient starts feeling better right away from for whatever reason, either because a symptom is removed or because the sort of bad effects, the systemic effects of a large tumor have now gone away. Yeah, so it's a relatively benign therapy.
How long is the light exposure, depending on the size?
Yeah, it's not very long. It's about 5 minutes of light exposure. And that's just to make sure that we get everything that, we overdo it. So, the light itself is non-thermal. You can't even feel it on your skin. So it doesn't burn anything. There's no burning at all. This is a chemical reaction that occurs. And it's induced by the light. So there's no harm in shining it for 10 minutes or 15 minutes, but all you seem to need is 5 minutes of laser light. Yes, sir. Yeah, so the question is, why did we choose the head and neck tumors? And so that's actually a really good question. And, you have some sympathy for drug companies when you're in this position because, there are all these cancers, all deserving of something, and you can see different ways of getting there, and like, which is the best one to choose when you don't even know what's going to happen. So there's sort of a risk in deciding what to do. But we felt that, number one, head and neck cancers are known to be very high EGFR expressors, especially those that are associated with human papillomavirus. So the cetuximab is an EGFR antibody. So we had a good shot of getting that antibody to the tumor because of the head and neck. Secondly, head and neck cancers, as you can see, are relatively exposed. So you can approach them through the mouth, you can approach them through the skin, but we end up having to use the catheters too. But still, you can see what you're doing. It's not deep inside the body. And third, the ear, nose, and throat doctors that we chose to help us with the trial had some familiarity with photodynamic therapy. So they had prior experience with lasers and putting catheters in and things like that. So they were sort of an educated group of ENT docs. Now, I have to say they were a special group of ENT docs because not all ENTs do this kind of work. But, one guy knew another guy, and you could get 5 guys who were pretty experienced in this. And that was our core of pioneers who got this off the ground. And I will say that now it's in Europe, United States, Japan, Singapore. The trial is going on. It's probably going to be approved in Japan first in next year because they have a more rapid approval process than we do. So
it would seem to me that it would be most effective in primary tumors, but not in patients with lots of metastasis.
Yes. And but you don't get to test a new therapy, especially this, in where it's considered that treatment is well established. You only get to test it in patients who have no other option. So the idea, this is a terrible disease, head and neck cancer, because chemoradiation results in, very dry mouth, bad dental problems, swallowing issues, speaking issues, a lot of really bad things. So if you could even move this therapy up to that, it would be a big deal. And we're now considering, where do these cancers come from? They come from mutations that start fairly early in something called premalignant lesions. So they're also known as leukoplakia. So the dentist, a good dentist, when he's doing your annual dental exam or semi-annual, will take a look in your mouth for white plaques, leukoplakia. And those can be premalignant lesions. Now, the trouble is you don't really know until you take it out. And you can imagine doing surgery in the wall of the mouth. It's a very delicate problem. And nobody likes doing that kind of surgery. And at the worst is you could put somebody through surgery and it may not actually be malignant at the end of the day. But if all you did was PIT, and they do express EGFR, then you could stop the tumor at that point, even before it's a primary. So it could be a preventive. And frankly, it could be put in the hands of dentists, honestly, because it's not a technically difficult. So, we're very excited about that whole process of moving it up. I'm personally thinking about prostate cancer, because current prostate cancer therapies are pretty radical. So it's either radiation to the pelvis, which causes bladder rectal problems, or surgery, which causes erectile dysfunction and incontinence. Major side effects. Yet many of the cancers that are removed are of questionable biologic significance. So the patient pays a big cost to become cancer-free. So why not do an antibody against prostate-specific membrane antigen, which is expressed by prostate cancers? Instead of ablating tumors, go in with the light as if you were doing a biopsy, but put a fiber optic, ablate the cancer, and treat it that way without disturbing the nerves or the urethra or any of that. So yeah, there's a lot to do.
Well, thank you, Pete. It's very exciting.
Yeah, thank you. Thank you for allowing me to change the subject.